Welcome to Medic's Desk. Today we'll be covering about high dose of methotrexate. In the previous lecture, we have already covered about the introductory part and the general side effects of methotrexate. Today we'll be focusing more upon the high dose of methotrexate. Methotrexate was introduced in year 1948 and it is the first drug which has shown a complete cure when used as monotherapy. It has got a broad range of therapeutic action. Methotrexate is divided into high dose, intermediate dose, and low dose methotrexate. High dose means more than or equal to 500 mg per meter square, and they are used in treatment of ALL consolidation, CNS prophylaxis of high grade lymphomas, leptomeningeal metastasis, primary CNS lymphomas. At the end of treatment, they require leucovorin rescue. Intermediate dose are divided into 50 to 500 mg per meter square and used in gestational trophoblastic diseases. Low dose methotrexate are less than 50 mg per meter square doses and used generally in CA breast, bladder carcinomas, and in various immune. Methotrexate enters the cell via three processes. The first process is an energy dependent process and it involves a carrier protein that transports folate and leucovorin into the cell. The second process is a passive process and is due to extracellular deposition of methotrexate causing a passive diffusion of the methotrexate inside the cell. The third process is endocytosis, which is mediated by overexpression of malignant folate binding proteins causing entry of methotrexate into the cells. To understand the mechanism of action of methotrexate, it is very important to understand folic acid metabolism. So, when folic acid enters the body, it is converted to dihydrofolate, which in turn is converted to tetrahydrofolate with enzyme dihydrofolate reductase. This tetrahydrofolate is converted to 5-methyl tetrahydrofolate and with the help of thymidylate synthetase, 5-methyl tetrahydrofolate is converted to dihydrofolate. I'll repeat it again. Folic acid is converted to dihydrofolate. Dihydrofolate with the help of DHFR is converted to tetrahydrofolate. Tetrahydrofolate is converted to 5-methyl tetrahydrofolate with the help of thymidylate synthetase. 5-methyl tetrahydrofolate is converted back to dihydrofolate. So thymidylate synthetase has got one more action. It converts DUMP to DTMP, which in turn forms thymine after multiple steps and is essential for DNA synthesis. So 5-methyl tetrahydrofolate further undergoes reduction with the help of methylene tetrahydrofolate reductase and forms 5-methyl tetrahydrofolate. This 5-methyl tetrahydrofolate with the help of methionine synthetase and B12 forms tetrahydrofolate. Methionine synthesis also has got a role in conversion of homocysteine to methionine, and this methionine is again converted back to homocysteine. This part of the cycle is called as methionine cycle. So we need to understand how exactly methotrexate acts. So methotrexate on entry into the cell, it inhibits dihydrofolate reductase. So there will be no conversion of dihydrofolate to tetrahydrofolate. It also inhibits thymidylate synthetase. So there will be no conversion of DUMP to DTMP. Hence, there will be accumulation of DUMP, there will be less DTMP, there will be no thymine synthesis and no DNA synthesis. The methotrexate also has an action on an enzyme which is called as amino iminodeazole carboxamide transformylase, which is also called as AICARFT. Also on glycemide ribonucleotide transformylase. These inhibit adenine and guanine synthesis and there will be no DNA synthesis. Due to inhibition of these enzymes, there is increase in the amount of AICAR and it acts like a anti-inflammatory. It has got an anti-inflammatory action. Hence it helps in other immune disorders. So just to remember, the mechanism of action of methotrexate is by interference of folate metabolism. The key enzyme that is targeted is dihydrofolate reductase. The other enzyme that is targeted is thymidylate synthetase as well as GARFT and AICARFT. Due to increased AICARFT, there is anti-inflammatory action into the body. cycle, which is divided into interphase and metaphase. Interphase consists of G1s and G2, and the other is metaphase. Let us see G1 phase. G1 phase lasts for 10 to 11 hours, and the cells of the body are metabolically active. They produce proteins, lipids, and carbohydrates. Also, organelles like mitochondria and endoplasmic reticulum. There are some checkpoint restrictions into this phase, which decide whether the cell is going to go to the next phase or is going to stay in same phase. Then comes G0 phase. It's a quiescent stage of the cell cycle where few cells will get arrested, like the mature neurons and skeletal muscles are always in G0 phase. The rate of RNA and protein synthesis also reduces. Then comes the very important phase, which is called as S phase. It lasts for 8 hours in human beings, and the DNA replication occurs here. Histones are also produced. Then comes G2 phase. The G2 phase, it lasts for 4 hours. The cell growth continues by protein and organelle production. The mitochondria also divide here. Tubulins are synthesized and the microtubules are formed. There is formation of something that is important, which is called as MPF, maturation protein factor, which helps in condensation of chromatin into mitotic phase. Then comes the mitotic phase, which consists of prophase, metaphase, anaphase, and telophase. Methotrexate, on entry into the cell, it inhibits dihydrofolate reductase, and it has got a high affinity than naturally occurring dihydrofolate. There is cessation of thymidine and DNA synthesis, ultimately causing cell death. As all these events takes place in the S phase of the cell cycle, it is called as cell cycle specific drug. Dihydrofolate is in great excess in a normal cell to provide reduced folate. Hence, the most commonly the cells which are affected are the neoplastic cells. Tetrahydrofolate synthesis continues until there is more than 95% cessation of the DHFR activity. To understand the further mechanism of action of methotrexate, we need to understand what is importance of polyglutamation. So, an enzyme polyglutamate synthetase, it adds multiple glutamate residues to carboxyfolates, which increases intracellular folate pool. And as this occurs, there will be accumulation of lot of folates into the cell, and they will not be excreted out of the cell due to bulky size and their charge. Methotrexate on significant concentration undergoes polyglutamation, and this can be achieved when the cells are exposed to drug for minimum 6 hours with a concentration of 2 micromoles per liter, which can be achieved only by giving high dose methotrexate doses. We need to understand how methotrexate polyglutamation helps us. So, intracellular accumulation of methotrexate will cause a reduced efflux of the drug outside, and hence there will be enhanced DHFR inhibition. There will also be inhibition of thymidylate synthetase, AICART, and GARFT. Hence, there will be inhibition of thymine and purine synthesis. The TS inhibition will also cause increased dump DUMP and reduced DNA synthesis and repair, ultimately leading to cell death. Till now, we have seen the benefits of methotrexate polyglutamation, but this polyglutamation is also responsible for the side effects of this drug. If the excretion or the leucovorin rescue is beyond 36 hours, there will be increased side effects. More the plasma concentration and longer the exposure to the drug, the polyglutamated methotrexate will increase in number. When polygglutamated tail has a glutamic acid residue of more than 5, the affinity for dihydrofolate reductase and thymidylate synthetase increases, and degradation and diffusion out of the cell is also slower. Variability in polyglutamization between tumor cell and normal cell explains why leucovorin selectively rescues normal cell from the effects of methotrexate but not the tumor cell cytotoxicity. Also upregulates the mitochondrial enzymes involved in the metabolism of serine and glycine, and it may also influence tumor cell sensitivity to methotrexate. Hence, involving the mitochondria, there will be altered energy production into the cell. It is very important to understand the rationale of leucovorin rescue to prevent the toxicity of the drug. Leucovorin is known by various names like folinic acid, N5-formyl tetrahydrofolate, or citrovoram factor. In 1950, Goldin and his co-workers, they did an experiment And after giving methotrexate, they provided reduced folates to bypass the metabolic block which was induced by methotrexate. In his experiment, he administered leucovorin 24 to 36 hours post methotrexate, and he was able to prevent the toxicity without reducing the tumor activity. The exact mechanism by which leucovorin helps in rescue of the cell is poorly understood. The deficient folate transport in the tumor cell may give us a clue that why tumor cells are not rescued by leucovorin as compared to normal cells. Leucovorin it competitively inhibits methotrexate and reactivates purine and thymidine synthesis. Polyglutamation in normal versus tumor cell explains why the tumor cells are more affected as compared to the normal cells by a high dose of methotrexate. The leucovorin intracellularly competes only with free methotrexate and not with polyglutamate methotrexate. We also need to understand that there can also be resistance to the methotrexate treatment. Why does this happen? Is it can be due to decreased transport of methotrexate in the cell, or there can be increased dihydrofolate reductase activity due to the gene amplification, or there can also be mutation in DHFR proteins which causes decreased affinity of methotrexate, or there can also be decreased polyglutamation process. There can also be decrease of thymidylate synthetase activity itself. All these can cause resistance to methotrexate. What are the lessons that we have learned today? Is that methotrexate is an anti-metabolic drug. It is a cell cycle specific drug. High dose of methotrexate undergoes polyglutamation. Polyglutamated methotrexate stays in the cell for a long time and it also causes multiple side effects. Leucovorin rescue is an important step for reducing the side effects. We have also seen. Thank you everybody and have a great day.